Okay, so now our job is to get that food that we have begun digestion on in the mouth and then in the stomach, get that moving into the small intestine where we can get some activity on it by the digestive enzymes that the pancreas is squirting into the small intestine. So let's get started, and then, oh, by the way, after that we follow it right on out the body. So here we go. So the small intestine it's a lot of tube. It fills much of the peritoneal cavity there in the abdominopelvic cavity because it is more than 20 foot long in the average adult. Its function is to complete the digestion of food and then to do the bulk of nutrient absorption. So approximately 90% of nutrient absorption will occur through the length of the small intestine. Only a little bit of absorption, primarily of water and of some vitamins that are being made by our hosts or our guests in the large intestine, make up that other 10%. All right, so the first 10 inches of the duodenum, of the small intestine, is referred to as the duodenum, and that's how you say it. It's duodenum. Although it was funny, I was just at a conference last week and was talking with a colleague there, and she said that she was doing some research on the proper pronunciation of this word and found that it is appropriate, that there is a secondary pronunciation of duodenum, but I think you will find that most people out there in the real world call it the duodenum. It receives the acidic chyme as well as all the digestive enzymes from the liver, and it also gets that bicarbonate ion that the pancreas is producing, and it also secretes an alkaline mucus. So between the bicarbonate ions and the alkaline mucus that's being produced here at the duodenum, the acidic chyme is fully neutralized, and thanks to the products of the acinar cells in the pancreas, we're continuing digestion of the food. The second section is referred to as the jejunum, and this is where if the duodenum is receiving all of these pancreatic juices in the jejunum, the mixing and chemical digestion is the biggest function there, and lots of the nutrient absorption occurs in the jejunum, and then the ileum completes the absorption that the small intestine is responsible for. And so here we are at the proximal end with the duodenum and the pyloric sphincter. Here at the distal end of the small intestine, it ends at what is referred to as the Ileocecal Valve. And let me just say this right now as well. Note the spelling of ileum. This is not the spelling that we used for the bone, so it is a different spelling. Note it, because many students will get confused on the exam. So we refer to the valve here as the ileocecal valve because ileo clearly is for the ileum. Cecal is because the very first portion of the large intestine is referred to as the cecum, and the cecum receives the waste material and begins to draw water out of it, compacting it into fecal material. So the ileocecal valve then, under, think about it, which kind of autonomic control? Would it be sympathetic or parasympathetic? Think. You can pause for a moment if you need to. Hopefully you said parasympathetic stimulation. So under parasympathetic stimulation, then the ileocecal valve will open, metering out the fully digested and nothing left but waste materials into the large intestine. So how does the small intestine remain in its location? Because it's filling quite a space from the top to the bottom, so most of the lumbar region is filled with, and a little bit of the thoracic region is filled with small intestine. What is it that keeps the top small intestines hanging up there where they belong? And that is a structure referred to as the mesentery. So if this is the tube looking down the pipe of the small intestine, the mesentery, the visceral peritoneum, it arises here. So we have parietal peritoneum at the wall, at the back wall here of the abdominopelvic cavity. Good night, I couldn't come up with that. The visceral peritoneum arises, surrounds the small intestine, and then returns to the parietal peritoneum. So this double fold here is referred to as the mesentery, and it suspends the intestines from the backside of the abdominal cavity. It also provides a pathway through which, so it's all connective tissue with a light layer of epithelial tissue on the surface, and that provides a pathway for bringing blood supply and nerves and lymphatic supply to the small intestine. And then at the very front, there is another double fold of visceral peritoneum. It is not attached to the wall of the abdominopelvic cavity, so it is definitely visceral peritoneum, and it forms what's called the greater omentum. And people describe it as sort of an apron-like structure. And if you choose to dissect a cat at the end of the semester, our cats are mature and the greater omentum is fully developed, you'll see how it just drapes across the peritoneal cavity. And this has a couple of functions. It's fat storage is one of them. So when men and women get that hard fat that is underneath the muscle wall of the abdominal cavity, that's because part of that hard fat is the greater omentum filling with fatty adipose tissue. But it also has a function of if an infection occurs, say here in the small intestine, we hope that it won't spread throughout the peritoneal cavity. The greater omentum will lay across it and can help to seal off that infected region and prevent the spread of infection throughout the peritoneal cavity. Okay, so let's look now at the intestinal wall. It has all the layers that we associated with the alimentary canal: the mucosa, the submucosa, the muscularis, and the visceral peritoneum. But on the interior, the mucosa is thrown up into villi. So we've talked so far about microvilli. What we haven't talked about before is villi. Villi are finger-like projections of the mucosa up and out into the lumen of the small intestine. And they are built of cells that are pseudostratified columnar epithelium that have on their surface microvilli. So this creates a huge surface area. I've never tried to calculate it myself, but I've been told that it is the surface area of the small intestine is approximately the same surface area as a tennis court. So if you can imagine that, if you took all of the villi and spread them out, and then further spread out the microvilli, that's how much surface area we have. That gives you an idea, at least, of what the surface area there is like. And so this increased surface area really facilitates the absorption of all those digested nutrients that are passing through the small intestine. Each villus has a lacteal. Remember, that's the lymphatic capillary that will carry away the digested and then reassembled triglycerides. We'll talk about that in just a minute. And the capillary network is there to take away all the other digested materials. So the surface of the, and here we're seeing the surface of a single cell. This is an electron micrograph, so super high magnification. That apical surface is decorated with microvilli that further increase the surface area. And because they get damaged as materials are passing through the digestive tract, these microvilli wind up being replaced every 3 to 6 days or so. Now, there are secretions that are produced by the cells of the small intestine themselves. We've already talked about the alkaline mucus that they make, but these cells also produce a large volume of very watery fluid that they flush out into the digested food material and then quickly reabsorb back into the body. So we have this constant flushing, reabsorption, flush, reabsorption. And what this winds up doing for us is that it's like doing the laundry, because you start the washing machine, you put a lot of water in there, and then you swirl things around, and then you draw that water out, carrying away the waste, all the dirt that was on your clothes. That is what's happening here, except what we're carrying away is not waste material, but it's actually the stuff that we want to bring into the body. These cells also produce enzymes, but rather than secreting the enzymes into the fluids that are inside the gut, they instead hold those enzymes on the membranes of the apical surfaces of the epithelial cells. And we see peptidases there. These, again, will further digest the smaller proteins or the amino acid strings into the individual amino acid subunits. They take and digest 3 disaccharides. These are 2 sugar molecules. Sucrose is a disaccharide built of glucose and fructose. Maltose is a disaccharide of 2 glucose molecules, and lactose is a disaccharide of glucose and one that you may never have heard of called galactose. That's one of the milk sugars. So these 3 disaccharides are fully digested, fully digested. So we break apart glucose from fructose, or the glucoses from each other, or the glucose from the galactose, thanks to the action of sucrase, maltase, and lactase. We know these are enzymes because of their A's ending, as we've talked about, and the beginning term tells us exactly what they digest. So lactase digests lactose, maltase digests maltose or malt sugar, lactose is milk sugar, and sucrose is plant sugar. So that's the sugar that we eat, table sugar, if you think about that, or cooking sugar that comes from sugar cane or sugar beets. And then finally, attached to the surface of the epithelial cells is intestinal lipase, which will further digest or continue the digestion of the fatty acids from the glycerol molecules of those triglycerides, the dietary fats. So how do we then get these materials in the digested material from the gut tube and into the body? Well, monosaccharides, so the glucose, fructose, and galactose, are all absorbed into the villi by facilitated diffusion or active transport. So facilitated diffusion, remember, no ATP required, but carrier proteins needed. And active transport is carrier proteins that use ATP. Amino acids are absorbed into the villi by active transport, so carrier proteins plus ATP. The fatty acids, because they are lipids themselves, are lipid soluble, and so they just diffuse directly into the epithelial cells. But in a crazy bit of work that our body does, we resynthesize them into triglycerides. So we take glycerol and we add the fatty acids back to the glycerol, and then we wrap that up into a protein package, producing a structure called a chylomicron. So a small bit of something. You have heard of chylomicrons before, but you probably may not have known it. They are the LDLs, the VLDLs, and the LDLs, HDLs, LDLs, and VLDLs the triglycerides that are carrying that are being carried in protein packages in the blood. And so when we look at when we do a blood panel, then we're looking for these chylomicrons as part of counting triglyceride levels. Electrolytes are absorbed back into the body by diffusion and active transport, and then water moves primarily by osmosis. So there you have it. That's how we get all these nutrients back into the body. Next is looking at the control of gastro, of the digestive system, of the intestinal movements. So parasympathetic stimulation causes the segmentation, mixing movement, and the peristaltic movements both. When the stomach wall gets distended as food fills it and the stomach wall stretches, that's what I mean by distension. This is stretching of the stomach wall. That triggers something called the gastroenteric reflex, which then automatically triggers an increase in peristalsis in the gut below. So let's think about this for a minute. I don't know if this is your experience. It certainly is mine. After I have a really big meal, one of the first things that I feel is an urge to go and void my bowels. So if you are noticing that same thing, what you're recognizing is the gastroenteric reflex. So as chyme fills the duodenum, that also causes stretching, and smooth muscle responds to stretching with contractions, so that increases intestinal peristalsis as well. And if the wall of the stomach is over distended, or if it's really irritated by whatever foods have been eaten, then there can be a peristaltic rush that will push all the contents out of the large intestine, or from the small intestine into the large intestine quickly, stretches the large intestine, and all of that just comes flooding out in the result of diarrhea. The gastroileo reflex will then cause relaxation of the ileocecal sphincter. So again, stretching of the stomach, that's the gastro part, causes relaxation on the iliac side of the ileocecal sphincter. Okay, so now we've moved our formerly food material out of the small intestine, and it is now wastes, because once it gets to the large intestine, there's not much reabsorption of nutrients that we can do here. So we see reabsorption, though, of water, because if we were to lose all of the water that's in the chyme that comes out of our stomach and into the small intestine, and if we were to lose that water that we're flushing through the small intestines, then we would become seriously dehydrated, as anyone who has had a lot of diarrhea probably recognizes. So we reabsorb water, and we compact the intestinal contents into the material we describe as feces. There is also a lot of absorption of vitamins that have been produced by the intestinal bacteria that live in there. So E. coli, which if it were in the stomach or even in the small intestine, E. coli would be a pathogen, but in the large intestine, it's a friendly little bacteria that makes a lot of vitamins for us. These bacteria also can make use of the cellulose, the dietary fiber that we've eaten, that is indigestible to humans, and so they will snack on that stuff, and along the way they produce for us, very thoughtful of them, intestinal gas. And then the last function of the large intestine is the temporary storage of fecal material prior to defecation. Okay, so let's look at the anatomy. The cecum is the first part. That's the expanded pouch that initiates compaction of feces, and it's also the site of the appendix with its lymphatic nodules that's there doing immune surveillance for us, checking out the food that we ate, making sure that there's nothing that we need to respond to immunologically. The colon is the next section, and there are 4 parts to the colon. The ascending colon, which goes across the right up the right side of the peritoneal cavity. Ascending because the food has to go, or the waste material at this point, has to go upward. The transverse colon, which moves across the body. The descending colon, which comes down, and then the sigmoid colon, which makes a little bit of an S shape as it proceeds first toward the posterior of the abdominopelvic, or actually down here it's the pelvic cavity, and then turns to come forward or toward the anterior side of the pelvic cavity, and then that descends into the last pouch for storage, which is the rectum. The rectum is found here at the end of the large intestine, and it can expand to store feces prior to defecation. So this is where the last stop before making the way out the body. The anal canal then passes through the body wall, and it ends at the anal sphincter here with 2 bands of circular muscle. The internal anal sphincter is a smooth muscle, and so it's under involuntary control. So parasympathetic stimulation causes it to relax, as does reflexive action as fecal material begins to pressure, cause pressure on, from the anal canal side of the sphincter. The external anal sphincter is a skeletal muscle, and so this is the muscle that we train. It's voluntary, and we train it to remain contracted until we give the signal, conscious signal, to open the external anal sphincter. But if there is sufficient pressure against the internal anal sphincter, causing it to relax, and then that then pressurizes or puts pressure against the external anal sphincter, at some point we will lose our ability to, our voluntary control of it, and it will involuntarily relax. And that's why, as we're running across the room trying to get to the commode when we have diarrhea, we sometimes have accidents. So defecation then is the movement of waste material from the large intestine by peristalsis. And rather than the large intestine doing peristalsis all throughout the day, instead it tends to occur, the peristalsis in the large intestine tends to occur in what are termed mass movements. So this is the 2 to 3 times daily movement of the large intestines causing the bowel movements. And frequently these follow meals because of the reasons that we've talked about before. The gastroiliac reflex, which is opening up and moving food into the large intestine, and so all of that additional bulk in the large intestine stretches it. So there we go. It helps to initiate a mass movement. And so, depending on the person, there are 2 to 3 mass movements a day. Folks who eat a diet very high in fiber can have more. Folks who eat no fiber at all may have many fewer, and sometimes have only a bowel movement every couple of days. So that is not considered normal. I think having at least 1 bowel movement a day is what's considered normal bowel hygiene. So as the rectum becomes distended with feces, then stretch receptors trigger the reflex that relaxes the internal anal sphincter. Conscious effort relaxes the external anal sphincter, and then with some peristaltic contraction, feces can be eliminated. Ta-dum! And there you have it, everything you wanted to know about the large intestine and the rest of the digestive system, but were afraid to ask. So, if it turns out that I was incorrect when I said that, and that there is more that you want to know, you know how to reach me. Find me during office hours. I wish you good luck studying, and we will see you next week.